Friends, I am Professor M.D. Kharia. I have retired from the service of Dr. Sir Hari Singh Gaur Central University. I have served the university for nearly 40 years in different capacities. Now today I am here to present my lecture on tannins. My dear budding pharmacist, as you are well aware of the fact that plant kingdom is the biggest factory in the world to produce phytoconstituents. These phytoconstituents are used for a variety of purposes. These constituents are mainly of two types, namely primary constituents and secondary constituents, where amino acids, fatty acids, and simple sugars such as glucose, etc., are primary metabolites. The alkaloids, glycosides, volatile oils, carbohydrates, resin, and tannins are secondary constituents or secondary metabolites found in the plant. Here it is worth mentioning that as secondary metabolites possess a varied type of biological activities, they are important source of medicines or drugs. In my today's lecture, I will be discussing tannins, which is one of the important category of secondary metabolites used in medicine in one or other way. Under tannins, I will be talking about their definition, occurrence, their medicinal value, general properties, and classification of tannins. Subsequent to this, I will take up some individual drugs belonging to tannin category. Now, I will be discussing the tannins under different modules, and under first module, I will discuss the general considerations about the tannins. Coming to the definition, tannins are non-crystallizable complex polyphenolic compounds widely distributed in plants. They are so complex in their nature that they cannot be defined precisely in few words. However, they can be well understood by the following attributes or the characteristics. Now, coming to the characteristics of the tannins. 1. With water, tannins form colloidal solutions which exhibit an acrid and sharp twisting and astringent taste to the mouth. The second one, tannins are characterized by their ability to combine with proteins, including proteins of animal hides, and converting these hides into leather. Third one, the tannins cause precipitation of solutions of gelatin as well as of alkaloids. They form dark blue or greenish black soluble compounds with ferric salts. And next one, tannins produce a deep red color with potassium ferricyanide and ammonia. Subsequent to this, tannins are precipitated by salts of copper, lead, and tin, and also by a strong aqueous potassium dichromate solution. The plant compounds which follow the above characteristics are considered as tannins. Now coming to the occurrence of the tannins, means where the tannins are present in the plant. There are certain important facts about this. For example, tannins are widely distributed in plant kingdom. Second one, the tannin containing plants are found almost in all families of the plants. Third one, the tannins are mostly found in leaves, fruits, barks, stems, and wood parts of the plants and trees. And next one, the tannin contents in the immature fruits is in higher percentage than in ripe fruits. Now coming to the significance of tannins for producing plants or the trees. Why the plant do produce the tannins? What is the importance of the tannins to the plants? First one, in plants, tannins gets oxidized releasing the energy. Now this energy is utilized by the plants for its various functions which is needed for their metabolic functions. Second one, as tannins possess antiseptic properties, they prevent the damage caused to the plants by insects, pests, fungi, or by physical agents or physical agencies, and thus tannins are useful in the survival of the plants. Now coming to the medicinal or therapeutic significance or importance of the tannins. First one, the tannins precipitate proteins and are able to combine with them, rendering them resistant to proteolytic enzymes. Thus, tannins protect the breakdown of proteins by enzymes. Second one, the tannins when applied to the living tissues, they produce astringent action. This prominent property of tannins is the basis for their use and applications in medicines and therapeutics. Now, due to astringent property, tannins are used as medicine both internally as well as externally. Internally, tannins are used in certain gastrointestinal or GIT problems or GIT disturbances. Subsequent to this, externally, as astringent agent, tannins are used to treat skin abrasions and burns, forming a protective coat by precipitating proteins of exposed tissues and cells, and finally helping the regeneration of new cells and tissues, thus helping the overall wound healing process. Next one, the tannins react with heavy metals, alkalides and glycosides, forming their inert complexes. As these complexes, which are also insoluble in GIT, and thus tannins are highly effective in treating persons suffering from alkaloidal, heavy metal, and glycosidal poisoning when used as their antidotes. As tannin containing drugs precipitate proteins, internally they are used for the protection of inflamed surfaces of mouth, throat, and stomach. As soothing agent, tannins are a good antidiarrheal agent, and recent studies revealed that some tannins exhibit good anti-tumor potential and anti-HIV properties. Proanthocyanidins, a type of tannins present in many herbs, has beneficial effects on health such as preventing angiogenesis and bacterial infections of urinary bladder and urinary tract. The tannins also find applications as laboratory reagents. Because of their precipitating qualities, tannins are used as laboratory reagents for detection of gelatin, proteins, and alkaloids. Now coming to the classification of tannins. On the basis of whether they follow the Gold-Beater's skin test, the tannins are broadly classified into two groups as below. One, the true tannins. These tannins give positive test with Gold-Beater's skin test, and most such tannins have high molecular weight ranging from 1000 to 5000. The second one are pseudotannins. These are the tannins of this group do not give true positive response to the Gold-Beater's skin test. Tannins of this group possess molecular weight less than true tannins, that is, their molecular weight is less than 1000. Now coming to the classification of true tannins. Classification of the true tannins is based upon two aspects. One, they should follow the gold beater's skin test, and second one, the colors they produce with iron salts. This color is based on the number of the phenolic groups and nature of their attachment in the molecule. On this basis, these tannins are further classified mainly in two groups. One is catechol tannins and pyrogalol tannins or condensed tannins. Catechol tannins are also called as flavotannins. These tannins are also called as proanthocyanidins. These tannins are neither hydrolyzed readily to simpler molecules and nor they contain any sugar moiety. This group of tannins show following characteristics. These tannins on heating yield catechol. Further, these tannins on boiling with hydrochloric acid, insoluble red colored flovins are produced. This group of tannins form precipitate with ferric chloride solution. Now such tannins with bromide solution form precipitate. Example of catechol tannins is the kynotannic acid present in kyno and catechutannic acid present in black and Gambier catechu. Other examples of drugs containing these tannins are green tea, cinchona, cinnamon and wild cherry bark. Now coming to the another group of the tannins, that is pyrogallotannins. These tannins are also called as condensed or hydrolyzable tannins, as they are hydrolyzed by acids or enzymes. Tannins of this group exhibit the following characteristics. These tannins on heating yield pyrogallol. Second one, when boiled with hydrochloric acid, these tannins yield gallic acid or ellagic acids. And third one, they form dark blue or blue-black color when reacted with ferric chloride solution. Fourth one, these tannins do not form precipitate with bromine solution. An example of tannins of pyrogallotannin group are gallotannin present in nut gall and ellagitannin present in the fruits of myrobalan and in the bark and fruit rind of pomegranate. Now I will be coming to the properties and test of the tannins. As has been told in the beginning, tannins are non-crystalline compounds. The tannins are soluble in water by forming colloidal solutions. Third one, tannins are soluble in dilute alkalis, alcohol, glycerol and acetate, but generally they are only sparingly soluble in other organic solvents. Fourth one, in solution form, the tannins precipitate heavy metals, alkalides, glycosides and gelatins. Fifth one, true tannins give positive response with gold beater's skin test. And the next one, all tannins give precipitate with phenozone solution. The precipitate thus produced is bulky and often it is colored. Now the seventh is the test for the catechins. Catechins, which are a type of tannins, on heating with acids, they produce fluoroglycinol. Now friends, with this general background on tannins, I am proceeding on some important drugs belonging to this category. Under Module 2, I will be dealing with some important individual drugs possessing tannins. Under this, I will be discussing the black and pale catechu, amla, harad and baheda. And to begin with, I will be proceeding with pale catechu. First one is the pale catechu. This is also called as Gambier catechu. Now coming to the official source of the pale catechu, Gambier or pale catechu is prepared from leaves and young shoots of Uncaria gambier. This tree belongs to Rubiaceae, family Rubiaceae. Coming to the geographical distribution, pale catechu yielding Uncaria gambier is a climbing plant which grows in Malay, Sumatra, Borneo, and some parts of Europe. Now coming to the preparation of the pale catechu. Leaves and young shoots of the plant are harvested since the plants are 2 year old by coppicing method till the trees attain the age of 20 years. Further, young shoots and leaves are cut in small pieces and boiled thoroughly with water in special containers. After boiling, the young shoots and leaves are removed. The extract so obtained is further boiled to a thick consistency of yellowish green color. This hot mass is then poured in wooden trays for cooling. While semi-cooled, semi-solid, the masses are cut into cubes by wooden knife. Then cubes are dried in sun and dried cubes are packed in suitable containers for market supply. Subsequently, I will be discussing the description of the pale catechu. Now, pale catechu occurs in regular cubes of 2 to 3 centimeter or rectangular masses or in irregular broken pieces. Pieces are light in weight and dull to dark reddish brown in color externally. Cubes or masses of the pale catechu are friable and thus they break easily. Internally, the pieces are porous and pale brown in color. Now coming to the odor, the pale catechu has no odor. The taste of the pale catechu, initially the taste of the pale catechu is bitter and astringent, followed by sweetest taste. And for your kind information, and the taste is sweetest, especially after drinking the water. Now coming to the constituent profile of the pale catechu. Pale catechu contains high percentage of catechin and catechutanic acids. However, rubinic and japonic acids are also present in small amounts. The other constituents of the pale catechu are catechu red, quercetin and gambier fluorescein, which is a fluorescent substance. Now coming to the tests. For the pale catechu powder when warmed with chloroform and filtered in porcelain dish and the extract is evaporated to dryness, a residue of greenish yellow color is obtained. This color is due to the chlorophyll present in pale catechu, which comes from leaves and green twigs of the plant. And another test is Gambier fluorescein test. The alcoholic solution of pale catechu when treated with sodium hydroxide followed by addition of few drops of petroleum ether and the mixture is shaken and kept for some time, a green fluorescence is seen in petroleum ether layer, indicating that the test material is pale catechu. Now coming to the adulterants of the pale catechu. Friends, as pale catechu is costly drug, it is liable for adulteration and its important adulterants are mineral matter like clay, starch, some astringent extracts and ferric salts and Dutch gambier. And subsequently, I will be discussing the standards for the pale catechu which are important. Now under this, the ash value of the pale catechu should not exceed 5% weight by weight. The cold alcohol insoluble fraction should not exceed 15% and the water insoluble portion should not be more than 20%. Subsequently, I will be discussing the uses of the pale catechu. Friends, pale catechu is employed medicinally as local astringent in the form of lozenges and as a general astringent to check diarrhea. Now, subsequent to this, I will be discussing the module 3, and under module 3 I will be talking about the black catechu, which is also called as kach or katha, or it is also called as true catechu. Now coming to the official source, black catechu is prepared from heartwood of Acacia catechu and Acacia chundra. The plant belongs to family Leguminosae. Coming to the geographical distribution, trees are of 15 to 20 feet in height. The trees grow in wild in India and Burma, which is now called as Myanmar. And coming to the preparation of the black catechu, the trees are felled by cutting from near ground. Leaves and small branches are then cut aside. The bark and sapwood are also removed from the trunk and stems. And then dark red colored heartwood is obtained and cut into chips. Chips are boiled with water in earthen pots, decoction and prepared. This decoction is strained to remove wood chips, and decoction is further boiled with continuous stirring until it attains the thick syrupy consistency. When sufficiently cool, the thick extract is spread on Cassia fistula, that is amalthus leaves, lying in wooden molds, and extract is left for overnight for cooling and solidification. When half dried, the brick mass is broken or cut in suitable sizes and packed in containers for market. Now, I will be discussing the organoleptic characters of black catechu. Black catechu is brown to black in color. Its fracture is short and fractured surface is dull glossy in appearance and blackish in color. Now coming to the odor. The black catechu has no odor, but it possesses very astringent and sweetest taste. Constant profile of the black catechu. Friends, black catechu contains catechu tannic acid to the tune of 25 to 33%, aca catechin, or which is also called as acacia catechin, to the tune of 10 to 12%. The black catechu also contains catechu red and quercetin in small amounts. And now here it is important to note that aca catechin of black catechu differs from catechin of Gambier in chemical composition and melting point. And the second important note is that black catechu is completely free from chlorophyll and fluorescent substances, which is present abundantly in Gambier or the pale catechu. Now coming to the substitutes of black catechu. Substitutes of black catechu are pale catechu and mangrove kach. These are the common substitutes of black catechu. Subsequently, coming to the adulterants of the black catechu, as the production of the drug is less and its demand is more, therefore black catechu is commonly subjected to adulteration with clay, red soil, ferric hydroxide and starch, where chemical dyes are mixed to get the desired color of black catechu. Now coming to the test for the black catechu solution when treated with vanillin or hydrochloric acid, it gives pink or red color. Second one, black catechu solution when shaken with lime water, a brown color is developed which turns to red precipitate on standing. Now there is a test actually to differentiate the black catechu from the pale catechu. And in this, to the alcoholic solution of catechu, sodium hydroxide solution is added to make the solution strongly alkaline. To this solution, when petroleum ether is added and solution is shaken, now if the solution gives brilliant green color fluorescence, the test sample is pale or Gambier catechu. However, if no green fluorescence is developed in the solution, the test sample is black catechu. Now I am coming to the standards for the black catechu. Under the standards, its S value should not be more than 6% weight by weight, and water insoluble residue should not be more than 25% weight by weight. Further, the alcohol insoluble residue should not exceed 90% weight by weight. Now, I am coming to the another important aspects of the black catechu, and that pertains to the uses of black catechu. Friends, black catechu has local astringent property for which it is used externally for boils, skin eruptions and ulcers. Internally, it is useful in cough and diarrhea. Black catechu has good cooling and digestive properties, and it also has pronounced antacid potential. Subsequent to this, I will be discussing the module 4, and under module 4, I will be discussing a very important drug, that is amla. You must be knowing about the amla. This is a very common drug. This is also called as ambelica or Indian gooseberry. Friends, in Sanskrit it is known as amalaki. Coming to the official source of amla, amla consists of dried as well as fresh fruits of Ambelica officinalis, or the another name for the Ambelica officinalis is Phyllanthus ambelica, belonging to family Euphorbiaceae. Coming to the geographical source or distribution, amla trees grow wild in deciduous forests of India. It also grows in Sri Lanka and Myanmar. Amla is a small to medium sized tree of 10 to 20 feet of height. Now, it is important to note that in earlier times, amla used to be procured mainly from wild trees. However, presently, due to deforestation, and second one, due to increased demand of the drug, a fairly good supply now comes from organized cultivation of amla trees in different parts of the country. Coming to the organoleptic characteristics of the amla, as far as the shape is concerned, when fresh, the fruits are depressed globose, and amla fruits are very hard and smooth. However, on drying, the fruits becomes dark brown in color and rough in texture. Coming to the size of the fruits, amla fruits are 1.5 to 3.5 centimeter in diameter, and initially the amla fruits are green to light yellow, which on maturity becomes brick red and finally dark brown in color. Now, coming to the smell, amla fruits have no particular smell, and initially the taste of the amla is sour and astringent. However, afterwards it is sweetish. Now, coming to the chemical profile of amla. Amla fruits contain high percentage of tannins. They also have high amount of ascorbic acid, which is also called as vitamin C, yielding nearly 600 to 900 milligrams of the vitamin C per 100 gram of the fresh pulp. Along with that, it contains some minerals, pectins and glucose. Now, coming to the composition of the tannins. Tannins of amla comprise of gallic acid, ellagic acid, phylambelin and triglycerol glucose. Now, here it is important to note that over drying or prolonged drying of amla fruits in the open sun should be avoided, as it leads to loss in vitamin C content. Now, coming to the uses and applications of amla. Amla fruits are widely used in Indian medicine as ingredients of many valued formulations. The most important formulations are Chyawanprash, Avaleh, with many health benefits, and a value tonic with free radical scavenging activity, and another drug is Triphala, which is a good tonic and mild laxative preparation. Here it is important to note, and it is worth mentioning, that unlike many other fruits, vitamin C, that is ascorbic acid in amla fruit, is retained even after drying the fruits. This is due to the presence of tannins in the fruits, which retards oxidation and thus deactivation of vitamin C. And second one, amla fruits are also good source of some minerals such as phosphorus and calcium. Subsequently, Amla fruits impart sweet taste to mouth, especially if water is taken after chewing or consuming the amla. This property of amla is due to the presence of tannins which modify the taste buds. Now, I am coming to the uses of amla. Amla have anti-diarrheal and anti-dysenteric action. Amla fruits are useful in jaundice and other liver diseases. The fruits have the liver toning properties. Amla is effective in dyspepsia and anemia, where it is used with iron compounds. Further, amla fruit juice is useful in leukorrhea. The fruits possess diuretic, digestive, emetic, and anti-inflammatory properties. Amla is also used in the preparation of hair oils and shampoos. The infusion of the triphala is used as eye wash to improve the eyesight. Amla fruits are very useful in treating scurvy, which is caused due to the vitamin C deficiency. The amla fruits are also effective in controlling diabetes. The whole amla fruits, after treatment with lime water, are processed in sugar syrup to give a preparation which is called as murabba. It is a coolant formulation, especially used in summers. The fruit juice or the paste of the amla, when applied to cut or wounds, it stops the bleeding. Amla fruits are useful in indigestion. It is important to mention here that alcoholic extract of amla fruits have shown antiviral, antibacterial, and antifungal properties. It is not only that the amla fruits are useful and have the medicinal value, the amla seeds are also useful in pharmacy. They are useful in asthma, stomach disorders, and bronchitis. They also possess diuretic, laxative, and refrigerant, that is, cooling properties. Now, friends, after discussing amla fruits, I will be discussing the module 5th, and under module 5th, the drug which I will be discussing is Baheda. Baheda is also known as Balearic myrobalan, and in Sanskrit it is known as Bibhitak. In Hindi it is called as Baheda. Coming to the official source of Baheda, Baheda comprises of dried ripe fruits of Terminalia balearica. It belongs to the family Combritaceae. Coming to the geographical distribution, Baheda is a larger tree which grows in India and Sri Lanka. The trees are found abundantly in Madhya Pradesh, UP, Maharashtra, and Punjab. In Madhya Pradesh, the trees are found widely in Chandwara districts. Now, coming to the organoleptic characteristics of Baheda fruits. The fruits of Baheda are globular and ovoid in shape, and they are 1.5 to 3.5 centimeter in diameter. The Baheda fruits are leathery brown, however, sometimes they are brown black in color. They have no smell, but their taste is astringent. The surface of the fruit is velvety, like fine suede leather. It is irregularly wrinkled, exhibiting 5 distinct longitudinal ridges. The fruits possess prominent round scar of pedicel at one end. The fruits are hard and when broken show yellow to dark yellow internal color. Now, I will be coming to the chemical profile of Baheda. Baheda fruits contain high percentage of tannins ranging from 20 to 30%. It also contains fixed oil which consists of esters of palmitic, stearic, oleic, linoleic acids. Now, coming to the composition of tannins. The tannins of Baheda fruits comprise of gallic acid, ellagic acid, phylambeline, ethyl gallate. Now, coming to the uses of Baheda. The main therapeutic activity of Baheda is due to its tannins, which are present in high percentage in fruits. As these fruits possess astringent property and therefore they are used in the treatment of dyspepsia, dysentery and diarrhea. Bahira fruits are important ingredient of valued formulation such as Triphala powder, which is an important purgative formulation of Ayurveda. Now, fruits of Bahira are important tonic. They are a good laxative, brain tonic, antioxidant, and free radical scavenger. That is, they neutralize the free radicals generated in the body, which induces aging process in the body, and therefore the Bahira fruits are potential anti-aging agents. The fruits are also effective in the treatment of leprosy, dropsy, and fever. Here it is important to note that half-ripe fruits of Bahira exhibit purgative property. This action is due to the fixed oil, which on hydrolysis exhibit irritant purgative action. And second note that unripe fruits due to the high tannin content exhibit prominent astringent activity and therefore they are more effective in diarrhea and dysentery. As mentioned above, the Bahira fruits possess both purgative as well as constipating effects. This is a beautiful example of variability in drug activity which is based on maturity level of the fruits. As a result, the same drug exhibit two opposite therapeutic effects at different maturity levels. Now, I am coming to the next module, module 6. Under module 6, I will be discussing the harad. The harad is also known as myrobalans or chebulic myrobalan. In Hindi, it is known as harad, harar, and in Gujarati, it is called as hirado. In Marathi, it is called as hirda, and in Sanskrit, it is called as haritiki. Coming to the official source of harad, the harad comprises of dried, ripe, and fully matured fruits of Terminalia chebula, belonging to family Combretaceae. Coming to the geographical distribution, the harad trees are found in sub-Himalayan tracts from Punjab to Assam. However, some trees are also found in deciduous forests of the Madhya Pradesh, Maharashtra, Bihar, and Gujarat. Now, subsequently, I will be discussing the organoleptic characteristics of the harad fruits. Coming to the size, the harad fruits are 20 to 25 millimeter long and 10 to 20 millimeter broad. Coming to the shape, the harad fruits are ovate and wrinkled longitudinally. Fruits are hard and strong. Coming to the color, harad fruits are yellowish brown to dark brown in color and they are odorless. Coming to the taste, harad fruits are astringent and slightly bitter initially. However, the taste is sweetish finally. Now, I am coming to the standards for harad. The harad fruits have tannins to the tune of 25 to 40% and water soluble extract to the tune of 40 to 50% and a moisture to the tune of 10%. Now coming to the chemical profile of the harad, as told earlier, friends, harad contains high percentage of tannins ranging from 25 to 40% and thus tannins form main constituent of harad. They also contain fixed oil which comprises of esters of palmitic, oleic and linoleic acids. Some glycosides, especially the anthracene derivatives of glycosides, and some glucose and sorbitol is also present in the fruits. Coming to the nature of Bahira tannins are pyrogallol type of hydrolyzable tannins which on hydrolysis yield chebulic acid, chebulic acid, ellagic acid, gallic acid and D-galleryl glucose. Now here is important to note that during the maturation of the fruits, the amount of the tannins in harad decreases whereas acidity of the fruits increases. These changes may be due to the hydrolytic processes occurring during the maturation of the hurred fruits. Now, coming to the uses of the hurred or myrobalan is mainly used as a laxative, which is mainly due to its anthracene derivatives. Hurred fruits are stomachic, means they enhances the gastric activity. They also possess prominent tonic, astringent, and anthelmintic properties. Hurred fruit pulp is a good blood coagulant and therefore it is used to stop the bleeding. This action is due to the high tannin content in the hurred fruits. Hurred fruits are a remedy for the treatment of piles and external ulcers. They are also effective in eye problems. Hurred fruits are important ingredient of trifla, a valued Ayurvedic preparation. Hurred fruits are appetizer as well as digestant. They have anti-aging and immune stimulant action which is due to their antioxidant property. Now hurred fruits powder is used to prepare the gargles, to cure inflammation of mucous membrane of mouth and to treat dental caries. Fruit decoction is effective in bleeding and ulceration of gums. Roasted fruits of the hurred are used to cure cough. The hurred fruits have the potential to cure nearly all GIT related problems and overall hurred is a good general tonic. Here it is important to note that hurred fruits powder when taken with warm water exhibit purgative action and therefore they are used to overcome the constipation which is due to the predominant action of the anthracene derivatives. The hurred fruit powder when taken with cold water it exerts dominant astringent action due to its tannin content causing constipating effect and therefore they have been found effective in controlling the loose motions. Now I will be concluding on my lecture on tannins. Friends, in my lecture on tannins, I have discussed the tannins, their occurrence, their physical and chemical properties, and their general medical uses. Subsequently, I have also discussed the black and pale catechu, which are used internally and externally both to control diarrhea, cough, and skin eruptions. Then I have also discussed very important drugs, namely the amla, baheda, and harad. And as you have seen, these drugs possessing a variety of biological activities are used in resolving the problem of constipation, jaundice, diarrhea, and dyspepsia. Now further, it is important to mention here that owing to their antioxidant and free radical scavenging activity, which is mainly because of the tannins and the tannin containing drugs are coming in limelight for their toning action on body and brain and also as anti-aging drugs and phytoconstituents. So I am sure from these lectures you must have understood the various aspects of tannins and also that how important this class of compounds are in curing or controlling different diseases or disorders related to the human health. Thank you very much.